On behalf of TechBharat team, a very warm welcome to the excellent panelists that we have put together today. We're presenting really a very big panel. The panel will be deliberating on the topic biomedical imaging and artificial intelligence for precision medicine. Let me give a small outline of the topic. Precision medicine is on the forefront of new paradigms in medical care. Since understanding a patient's health and mechanisms is an arduous task, the use of artificial intelligence has become a crucial tool in understanding patients' genomics in order to generate tailor-made therapeutics. One of the highest potentials of AI is precision medicine in its use in preventing diseases using techniques that could assess the risk of disease development. Despite the excitement AI has generated in medical imaging research, there are challenges before it can become more robust and be widely adopted in the clinic. On the short term, two challenges need to be resolved before AI can be more widely implemented in medical imaging research. First, how to organize and pre-process data generated from different institutions. Second, on a policy or infrastructure level, how to encourage more image data sharing. Currently, image data sharing is very limited. HIPAA compliant is one concern and lack of infrastructure is another. The medical data security needs to work with the emerging needs of data sharing. Corresponding infrastructure also need to be built. To deep dive into these very, very sensitive issues, we have put together a great panel. Let me introduce the panel. Dr. Shiv Nagraj, Research Fellow, Senior Research Fellow, Queensland University of Technology. Mr. Rupert, co-founder of and CEO, Tissue Gnostics. Mr. Marshall, I think he's replacing Jay. Adarsh Natarajan, CEO and founder, and Mr. Amruta Kulasinghe. Dr. Kwan, Group Leader, Genomics and Machine Learning. Before I hand over to Dr. Shiv, a small introduction of Dr. Shiv. Dr. Shiv Nagraj is an internationally renowned scientist with expertise in computational biology and genomics. He is the Advanced Queensland Research Fellow at Queensland University of Technology in Brisbane and leads a team of multidisciplinary researchers. His lab's overall focus is on improving human health and applied genomics and artificial intelligence in cancer, transfusion medicine, and indigenous health. I now hand over to Dr. Shiv Shankar Nagraj.
Thank you, Shekhar. Thank you for that introduction. Hello, everyone. On behalf of TechBharat in India, I welcome all of you to the panel discussion. And we have a very diverse international team of experts today to talk about how biomedical imaging and artificial intelligence is an emerging and an important tool for the precision medicine. Shekhar did give a brief preview, but I would also like to make some initial remarks. So for me personally, it's a great pleasure to moderate this panel. And I think the timing of the discussion is perfect, given there are so many new developments in the field, both in the imaging and the genomics, with the new spatial technology coming into the picture. A lot of changes are happening. Imaging and omics in general, where I have been in the last 10 or 15 years, have seen that these two have historically been isolated fields. People in radiology really didn't talk much to the genomic folks and vice versa. And at the same time, we all know that pictures can speak a lot of words. So molecular imaging and this omic molecular imaging and the different omics technologies and the high throughput data that we are generating in genomics, transcriptomics, proteomics, and metabolomics are really creating opportunities on both sides to now start to talk to each other. And as we see, imaging technologies have opened up new directions in pathology. And as we can now study spatially resolved. Okay, so this is a big change, spatially resolved proteomic and genomic profiles. So I know that a lot of us work in the area of cancer and we are very focused on cancer. although some of the things that we discussed today is also going to be applicable to other diseases. And cancer is a great example, as you all know, that how the spatial bioimaging methods are helping to study tumor microenvironments, particularly in immunotherapy. Molecular imaging helps to track the side effects and downstream results of therapies. So this next generation of imaging technology is actually heavily supported by artificial intelligence. And we have all witnessed that in the last few years, increasingly with the deep learning models, etc. So AI-powered algorithms can be used to drive clinically relevant and actionable information. And we have done this in the cancer genomic space and transcriptomic space, and independently in areas in the radiology. But what is also emerging now is a new area called radiogenomics. And AI also has the potential for patient profiling that is data driven, and it enables the personalized medicine. My own lab has developed a new tool called Imagine that combines the radiomic images with the genomic data set. So we have multiple machine learning algorithms in the tool. So we are already seeing some fantastic results in glioblastoma and other cancer data sets. So with this rapid change that is happening in the last few years, I think it would be just a great time for this panel to talk about. So collectively, this panel carries a lot of experience in R&D, in product development, in translation, and entrepreneurship, right? Which means carrying discovery to the patients in both developed and the developing world. So without that as a remark, I would like to really start this. And what is more exciting for me to bring these panelists together is this opens up new channels for academic and commercial partnerships, not only among the panelists, but now sort of beyond the borders of different countries. I guess with that, I will start and introduce the first speaker for today. So the format of this conversation would be a brief talk, maybe 6 to 8 minutes by individual speakers. And after that, we'll open the floor for discussions and questions. So indeed, it's a great pleasure to introduce Dr. Rupert Ecker, who is our first speaker. He's a co-founder and CEO of Tissue Gnostic. It's a European-based company, mostly with a strong presence in Austria, Romania, and USA. He is a co-inventor of a product that they have developed within the company called Tissue Fax technology. And he has also headed several joint R&D projects in advanced computer vision, cancer research, stem cell biology, and personal medicine. So there can be no better person to start this kind of conversation than Rupert. And most importantly, his company, the digital pathology products that his company has developed now have a market presence in more than 60 countries in the world. And recently, we are very glad that he's a visiting fellow at QUT since last year. So it's such a great pleasure to introduce Rupert to give a talk. Rupert, it's yours.
Thank you very much, Chief, for the kind introduction. I'm just sharing my screen with you.
Please.
Okay. And welcome, everyone. It's my great pleasure to be the first speaker in this thrilling session among this panel of experts. And I am happy to give you a short introduction to my background and my company, which is focused on a technology that is tissue cytometry. Now, Shiv, as you said, we are working here with discussing the precision medicine, this mega trend into the 21st century, where we bring genomic, proteomic, and other omics data together. So there is a big amount of data that have to be incorporated, brought together. And when we are talking about data, we should maybe have a critical look on how we generate the data. And there has been this most astonishing publication in Nature a few years ago, which caused a big discussion in the community about reproducibility, or it was also called the reproducibility crisis. You may be aware of it, but I would like to bring the whole audience, the attention of the whole audience to it. A survey was published among 1,500 scientists, which actually led to the conclusion that more than 70% of the researchers have problems in reproducing published data, and another 20% even have problems in reproducing their own data. So the question is the reliability of our published data just 10%? The authors of this survey actually call for a better mentorship. This is something that universities have to take care of, proper use of statistics. This is something each individual research lab has to take care of, and more robust and experimental designs, which increase the reproducibility. And this is exactly where our company, Tissue-Gnostics, comes in. Who is Tissue-Gnostics? It's, as you said, a European company. It's based in Vienna, in Austria, the headquarter. We have subsidiaries around the world, Romania, USA, and in Asia Pacific. The company is licensed in Hong Kong and has offices in Beijing and Brisbane. And as you said, we are now have clients in more than 60 countries on all continents, and more than 1,680 reference papers have been published over the past 18 years since the company was founded. It has always been critically important for us as a company to work in close collaboration with high-end research in academia. And as such, we have been a partner in currently 5 European-funded innovative training networks, which dealt with digital pathology, the calcium sensing receptor as a potential target of therapeutic intervention, translational oncology, the cell network of cell migration studies, and currently on big data analysis for clinical benefit using machine learning approaches. We have for this, in order to support our clients, we offer instruments and software which automatically scan tissue slides and identify single cells in tissue section and allow to quantify markers, molecular markers in all types of tissues and in all types of disease. When we look on this sample, this shows an immunohistochemical staining of colon, a well-known marker in brown, Ki67, a proliferation marker, and the counter stain in blue is hematoxylin. We made, not among 1,500 scientists, but among 100 scientists, a survey and asked, what do you think, how many cells are proliferating? In other words, how many blue nuclei are brown? And we received answers ranging from 1 to 40%. And this reflects also the issue that I have shown in the first slide of the reproducibility. This shows the major problem which is associated with visual evaluation. Depending on whom you ask and even when you ask, early morning or in the afternoon when people are tired or when the sun is shining or it's nighttime, you may get different answers, which give you a range of estimations. With what we have developed, this is the TissueFax cytometry platform, we can analyze the samples automatically. You see here in yellow what the software has automatically identified as a single cell, and then quantify how much staining is associated with each cell. And what we get is this type of scatter plot, like in a flow cytometry readout, where each dot represents a single cell, but now in a tissue section. And with this technology, it is possible to quantify molecular markers in any kind of cell, on any kind of tissue, in any kind of disease, and thereby provide a basis for observer independent quantification and replace the observer biased estimations by real measurements. And one recent application that I would like to show you comes from a group at Harvard in Boston. In this study, which was published last year in Cell, they quantified the immune response against the SARS-CoV-2 virus in COVID-19 patients by using the TissueFax technology. So the quantification was done in situ, directly in the tissue, using a multispectral imaging approach, which is one of the technologies that we support. And 7 markers were stained on the same type of cell populations directly in the tissue and analyzed. And obviously, I don't have the time to go into detail here, but what this study has shown is that, unfortunately, the coronavirus, which we are all currently suffering from, escapes the immune system in a way that it prevents the formation of long-term immunity. So the concept of herd immunity, which many countries have relied upon at the beginning of the outbreak in 2020, this concept does not work. And here, based on this in situ data, we now understand why. A major trend, and this is the entire topic here for this panel today, is artificial intelligence. And this is also one of the topics that we have been working in over the past 10 years. And so to provide tools to the research community around the globe that allows to have such tissue, obviously this is skin, what we see here, assessed automatically, have it separated automatically into different histological entities, right? Like what is the epidermis, what is the dermis, what is the subcutis, and other structures, which is very labor intense, but also very important for a differential diagnosis that is required in precision medicine. Not only where, which cell is there, but also where is a certain cell population to be found. Because cells that are required, for example, for immune defense, may be in the wrong place in the tissue, so they still cannot act properly. And this is made to define the localization, the spatial distribution is made possible by such advanced technologies. Here we see an example of another 7 color staining of an immune checkpoint profile. The original image, and after spectral unmixing, we get a very clear, beautiful, although it's cancer, we can say it's a beautiful image. And then this allows a differential analysis on the individual subpopulations, with each dot that we see in these scattergrams representing a single cell in the tissue section. And based on such data, we can then really make a differential diagnosis where in the tissue are which cells and how do they act. And this is the basis of precision medicine, to understand the molecular mechanism, not on a statistical level, but on an individual patient-specific level, to quantify molecular markers. That means we need to quantify an ever-increasing number of molecules on the same subpopulation of cells. And our latest project that we have started is, we call it the virtual histologist. That is a system based on machine learning, which will allow to classify tissue sections. But this is not the new idea, but the new thing is that we work here, include here machine learning technologies of latest generation, which allows to teach computers how they can learn from other computers. Normally in machine learning, we start with a training set, teach the computer what is tumor, what is normal, or what is a glomerulum, what is a duct or whatever, and then the computer learns. But if we move to another sample, or another tissue type like prostate instead of kidney, then we start from the beginning. This is very different to how humans learn, right? When an expert, a medical doctor, a researcher, but also every person on this world is confronted with a new question, he does not go back to kindergarten and learn everything again, but builds on existing know-how. And this is something where we are now working to allow computers to learn from already existing machine learning models and not always to start from the very beginning. And with this short overview, I would like to close my presentation, and I'm definitely happy and looking forward to the panel discussion. Thank you very much.
Excellent. Thank you very much, Rupert, for that great overview and a lot of experience there in the area already. So it's my pleasure now to go to the second speaker, Adarsh Natarajan, and Adarsh is the CEO and founder of Aindira Systems. So it's based out of Bangalore in India. Adarsh has founded this about 4.5 years ago. His company is AI-driven company that is focused on creating diagnostic solutions for fatal illnesses. So Adarsh has a combination of background. He has a technology background, and also he has an MBA from a prestigious Indian institute called IIM Bangalore. He is a very regular speaker, and he has been invited by Harvard, Forbes, TEDx conferences. So he's a very active TEDx speaker as well. What he is trying to do in the Indian scenario is really develop something, a system, a combination of hardware and software in the imaging space and digital pathology space that is very, very important for point of care in the Indian context. So with that background, I will invite Adarsh to present his talk. Adarsh, over to you.
Thanks, Shiv, for the introduction and the kind words. Still, compared to all the eminent panelists, we're just stepping out of the woods. So thanks for having me speak about our experiences and our perspectives. So I'll just quickly start with, just give me a second while I share. Yeah. So yes, as Shiv was introducing, I'm the founder of Aindra Systems, and our mission is to save lives through early detection. So specifically, with the context of our discussion today, I'd like to focus on delving a little bit deeper on what are the characteristics that India as a healthcare market has, and therefore that would provide a better understanding and set the context for what we are doing as well. So just a quick snapshot, 850 million people, right? This is the presently the population of India that resides in the rural parts of India. So I'd like to let that number sink in. 850 million. We're talking about twice the population of US, and probably more than the combined population of many of the smaller European countries. So it's a huge population that resides in non-urban centers in India. Right, and that poses a separate set of challenges. It has its own dynamics. Now, combined with that, there are certain challenges that our healthcare system, the Indian healthcare system, has as well. So you have just one allopathic doctor for every 10,000 people, right? Um, we have one state-run hospital for 90,000 people, right? So now you kind of understand, with the 850 million people who are needing good quality healthcare systems, what is it that is available for them, right? This one allopathic doctor for every 10,000 people, one state-run hospital for about 90,000 people. Uh, Very dismal numbers. However, you see in all of India, the distribution of healthcare facilities, 75% of good quality healthcare systems cater to only 35% of urban India. And that's the kind of paradigm that we are talking about, right? So on one side, you have a huge population residing in rural India that is in need of good quality, accessible, affordable healthcare systems. But on the other side, you see that the urban centers are oversaturated. They're saturated with most of the healthcare facilities trying to cater to the same catchment of people, right? So this introduces very specific dynamics. Now, how the government system is set up, and I know this needs a little bit of understanding as well. So the healthcare model is a three-tiered model, which starts with a sub-center and then goes on to what is called as a primary health center and then proceeding on to a community health center. This is how the government healthcare system is set up here in India, right? So we have about 150,000 sub-centers. These are very basic healthcare facilities, which is manned by one, what we call as an ANM, or auxiliary nurse midwife. Essentially, just very basic mother and child treatment can be provided by these ANMs. The next PHCs, we have about 25,000 of them in the country, uh, which ideally should have one medical officer and about 14 paramedics. So this is the second level in the second tier. The third is followed with the community health center, which we have about 5,000 of them. And the ideal number is that it has to be catered by about uh, four medical officers and 21 paramedics. So this is how the government healthcare system has been set up, predominantly to cater to the 850 million people who are in the rural India. However, the reality, stark reality, unfortunately, is 10% of these PHCs, which I said about 25,000. So we're talking about 2,500 PHCs have no medical officer at all, right? And 20% of these PHCs have no pharmacist. Remember, I told you that they have to have about 14 paramedics, right? Of which a pharmacist is one of them. And 40% of these PHCs have no lab technician at all. So now you see the kind of need that is there on one side and what is the reality. And I'm specifically talking about the Indian healthcare ecosystem. I'm sure many of the other developing countries would be suffering from similar, if not exactly the same characteristics as well. So, what is the need of the hour from our perspective for geographies or countries that share these kind of dynamics is that we need to have point of care solutions, not really centralized large healthcare facilities, but more disaggregated, more distributed facilities, which have point of care solutions available for them. Uh, and they have to be affordable, not to speak about overemphasize this part, but it has to be over affordable for this large population to make use of. And of course, by virtue of being available in each of these primary health centers, private clinics, all of them put together, it makes it accessible to the larger population. So these are some of the imperatives that's a need of the hour, right? Now, when any of us, like entrepreneurs starting out on a journey, or even businesses trying to cater to this large vast population, underserved population, what we do need to keep in mind are some of the imperatives, right? And this goes to the point that I'm talking about, that all the solutions, all the problems that we are addressing should be able to work in low resource settings. Now, what do we mean by low resource settings, right? At least in India, we are talking about situations where there can be frequent power cuts. There can be very sketchy or negligible internet coverage. Human resources may be available, but they'll be very low skilled. And of course, the equipment and the expertise available to deal with that is also going to be extremely low. So all that we develop should be able to cater to these low resource settings, and that's what the context that I was talking about. Ease of use, right? When we're talking about ease of use, any of these solutions that we develop for this market has to be very easy to use, meaning that we are talking about having ANMs being able to use any of the solutions. It cannot be very highly sophisticated equipment requiring higher skill sets to even use them. So we're talking about extremely user friendly, high in terms of ease of use ability for the systems that we're developing. Not to overemphasize this at all, price is a huge determinant in wider adoption. So these are some of the, I mean, we talk about transplanting what is available in the Western world into context which requires a completely different treatment, and we suffer with some of the challenges that arise with that treatment. Meaning, right from, of course, requiring high resources to use that, we are also talking about price points that are pretty much unaffordable for these contexts. So these are some of the imperatives that any solution that gets developed keeps in mind. Right? These are the central keystones of those solutions. Coming back to what we are doing to address these issues. So as I kind of already mentioned, there is, on one side, we have an acute shortage of health professionals, uh, as evidenced in some of the numbers that I shared earlier. On the other side, what we have done is, unfortunately, because of importing systems that are not built, not designed, and conceptualized for a distributed healthcare system, unfortunately, our healthcare system also has come to become configured as more centralized rather than distributed. So that's kind of a dichotomy that we are dealing with. Now, the impact of this is, on one side, we see that if once these healthcare systems are centrally configured, more centralized than disaggregated and distributed, it inhibits accessibility to a great extent, and lack of accessibility drives up the cost. So affordability takes a hit as well. And on the other side, what we see is healthcare services are under pressure because, as I mentioned earlier, 75% of these cater to 35% of the population, meaning that they are fighting it out in a oversaturated market, right? So therefore, on one side, you see that there is lack of access to a vast population, and on the other side, because of the way the healthcare systems are configured, the existing healthcare services are also under pressure because they're catering to a smaller population, right? And there was a study done by in India where about 10,000 labs were sampled, and it was shocking to see that these 10,000 labs were run by only about 2,200 pathologists. So it's about one in five. So one pathologist for every five laboratories. So this is kind of shows us the magnitude of the impact that we're talking about, right? So the solution that we came up with was a computational pathology platform to address some of the issues. Of course, this is just not going to be a silver bullet that will solve the entire accessibility, affordability gamut, but goes towards doing some part of it, right? And this is a point of care solution. It's affordable and accessible. And our belief is that it can go a long way towards democratizing healthcare. So we've designed it in India, but pretty much relevant for many parts of the world as well. And I'll take a little bit of time to explain what the computational pathology that we're talking about. And this is kind of similar to what Rupert earlier mentioned about. So we are also in the imaging space, but what we've developed is a combination of a hardware and software solutions. Now, four components all working together to make this a point of care system, right? And keep in mind that all of these hardware components that we are talking about are very low footprint, low power requirement devices that can be placed on a bench top or even carried in a small mobile medical unit as well. So we have the first system, which is an automated stainer with a very low footprint one, which can stain biological samples once they're extracted from the patient. The second system is what converts that into a large digital format, which is a whole slide image. The third component, Astra, is the AI part, which analyzes the digitized sample. And all these three are all edge devices, meaning they're designed and conceptualized and developed to work in a low resource setting with very limited or very negligible amount of power or even internet settings. So all three of them are meant to be an edge computing device. Now, the fourth component, cluster, is the telepathology platform, which through which pathologists sitting across the limited number of pathologists that we have, we can dip into that pool. They need not be physically present in any of these centers, and this allows a kind of decoupling of the pathologist from looking at the samples through a microscope. So this in turn completes the whole end-to-end treatment for a computational pathology platform. And this is what we at Indra Systems have put together over the last few years that Shiv mentioned. Our first focus area is, of course, cervical cancer, and the problem that we've taken up because this is specifically very large in India, globally as well, where one woman dies every 2 minutes, but in India, we're talking about one woman dying every 7 minutes. So in India alone, we're talking about 460 million women at risk in India. Unfortunately, India has one third of all the global mortalities for cervical cancer, and pretty disturbing numbers. All because of non-availability of a reliable means of getting themselves tested early on. So the product that we've developed leveraging our computational pathology platform is called as Sarvastra, Serve for cervical cancer, and Astra, the underlying AI that drives it. What we've seen, and this goes back to my earlier point about accessibility, right? At the present moment, if a lady in a faraway tier 2 town or a tier 3 town in India gives her sample, cervical swab sample for testing, it takes anywhere between 4 to 6 weeks for her to get a report back. And this is something that is pretty disturbing, because we see that there have been studies which indicate that if the follow-on recommendation counseling does not happen immediately once a woman is active and is available in the center, the drop-off rates for any treatment options goes to about 50%. Right, so this is a very key problem as to why there is no compliance to recommended treatment options, and that is something that is a problem that something like a point of care system like ours will address very sweetly, right? So we give out results in under 30 minutes as opposed to what it takes because of all the logistics, collection, and then going back takes in the normal way. So we are also ISO 13485 certified, and of course we have higher sensitivities that we've seen compared to the manual way, which is about 65% sensitivity, according to numbers put out by IARC, that is International Agency for Research on Cancer, for detecting cervical cancer using pap smears. I'll just end with some, time to call it some of the support, some of the support that we've received. We've been supported by the Indian government, the US government as well, and multiple other partners who've helped us translate some of the research that we've done into a product that has a high social impact. And happy to also quote that we've been mentioned by the media multiple times. And with that, I'd like to kind of hand it back to Shiv, and happy to take questions as we proceed along.
Thank you very much, Adarsh. I look at your talk in both ways. One, the grim picture of the reality of the rural healthcare. But at the same time, we have entrepreneurs such as you who are trying to break this and create new optimism, which I'm very sure that we will have better health outcomes moving forward. So let's move to the third speaker. Our third speaker is Marshall Fateo from NanoString. So Marshall is a technical sales specialist at NanoString Technologies, and he manages the APAC region. He's an expert on spatial genomics. Marshall has a PhD in microbiology and immunology from James Cook University in the northern Queensland, and he transitioned into commercial side of life sciences with an emphasis on genomic technology. So all of us agree that NanoString is clearly market leaders in spatial genomics. And we have witnessed that in looking at the data, the instrument, the change in technologies in the last few years. So what we expect, and we're really looking forward, is Marshall is going to talk about this. He's going to talk about spatial technology and what's happening in this very exciting area. Marshall, over to you.
Thank you, Shiv. Just share my screen. All right. Yeah. Thanks again for the introduction, Shiv. And apologies for Jay Manakandan, who I'm filling in for. As I mentioned to some of the panelists earlier, he broke his arm yesterday, so he's currently at the hospital getting an MRI, and we all wish him a speedy recovery. So today, as Shiv had mentioned, I'm going to talk about sort of the landscape of spatial biology and more of a global sort of 30,000 foot overview for everyone. So if we consider the evolution of genomics research, we know that bulk methodologies, including NGS and high throughput gene expression profiling, similar to what our nCounter platform can do, have provided important discoveries in both health and disease. However, a limitation of bulk methodologies is that you have to homogenize the tissue. And when we do that, we lose cellular granularity. And that's with respect to what specific genomic signatures are created by which cell. And that's represented by the fruit smoothie here. So if we fast forward to 2012 and the advent of single cell RNA-seq technologies, which 10x Genomics, of course, has done an excellent job with, researchers are now able to profile single cells in sort of a fruit salad kind of context with using multi-omic approaches, so transcriptomic and proteomic approaches. And we know that these single cell RNA-seq initiatives have underpinned global atlasing initiatives like the Human Cell Atlas, which are attempting to identify and understand cell types in different organisms and model systems. And while single-cell RNA-seq is integral to understanding these cell populations, when you dissociate a tissue, you lose cellular context. And when you lose cellular context, that determines how cells are arranged in tissue, and that directly governs their behavior and how they communicate in both healthy and disease states. So spatial context allows us to interpret cellular ecosystems, heterogeneous cellular ecosystems, and build on the knowledge that we can glean from bulk and single cell technologies. So as proof that the field of genomics is adopting the use of spatial technology and moving into the future, Nature Methods dubbed spatial transcriptomics the method of the year for 2020. And so former recipients of this designation include NGS sequencing and also single cell RNA-seq. And spatial biology moving forward is going to really redefine and revolutionize how we understand tissue biology in both healthy and disease settings. So here's an example that we gave earlier. Rupert also mentioned this, but this is a tumor cell. And we know that translational and discovery scientists, we work with heterogeneous tissues, and there's a need for high parameter analysis in all of these spatial tools. And we know that a tumor and other tissues, of course, are comprised of an amalgam of different cell types. In this particular example for the tumor, we have our immune cell populations, and you have the tumor itself, cancer-associated fibroblasts, et cetera. And what cancer scientists and oncologists are interested in is the interaction between the tumor and the tumor microenvironment. So how are these immune cell types interacting with the tumor, and how does that reflect disease progression? So some of the questions that researchers are asking are, why do some patients respond to treatment and some others do not, particularly with immune checkpoint therapy? What is the mechanism of disease progression? In terms of biomarker discovery, are we able to identify specific signatures in different micro-niche compartments across the tissue that can help inform on therapeutic development and or treatment guidelines? And of course, what is the heterogeneity of those cell populations that are sitting within these tissues? So as you can see, the spatial revolution will take discoveries in the lab and ultimately translate these findings to improve patient outcomes. In this slide, you can see that there's 3 separate bins, one for discovery, translational, and clinical research. In the discovery setting, scientists are examining whole tissues and also informing on single cell RNA-seq work and specific cell types in those tissues. And they're taking more of a 30,000 foot view, trying to determine if they can identify specific signatures or patterns in those micro niches across the tissues. And that can be in either the mouse or the human, depending on the platform. And these analyses will give insight into clinical application. So from the discovery platform, moving into the translational space, scientists can systematically reduce the number of targets, or fine-tuning their approach based on their discovery platforms, and focus on those targets and samples that are collected directly from patients. So we know that FFPE is the primary sample type that we get out of clinic. So once the translational research is complete, scientists can then apply these discoveries and test their discoveries in patient cohorts. And an example of this is identifying specific biomarkers in patients that might help diagnose disease, predict disease outcome, or guide on potential therapeutic interventions or combinations thereof. So here's an example of that translational research in practice. So in this study, this is Dr. David Rim from Yale University, and he used our GeoMx digital spatial profiling technology to identify a specific signature in segmented compartments in non-small cell lung carcinoma. So if you look at the panel on the top hand right here, this is an image taken off of our platform. So what we're able to do is stain specific cell populations with fluorescently labeled antibodies. And we have a microscope embedded into our system, and we can use algorithms to segment these specific compartments and cell types based on those morphology markers or those fluorescently labeled antibodies. So in this particular example, in green, here's the tumor compartment, and then we have a mixture of immune cell compartments in yellow, purple, and blue, respectively. And what Dr. Rim was able to identify is that he found two biomarkers that were predictive of progression-free and overall survival in these cancer patients. And the power of using this particular technology is that he wouldn't have been able to identify this specific signature in the CD45 compartment had he used bulk or single cell methodologies or other spatial platforms for that matter. So here's another example. I have to laugh at this slide. So I put this together before I knew Arutha was going to be on the panel today. But his story is really a testament to how understanding the context and application of spatial technologies can really accelerate discovery and answer some important questions. So earlier this year, or last year, excuse me, Arutha and his group, they wrote a couple of review papers outlining the importance of the tumor microenvironment, of course, and also in the application of spatial technologies and defining that, and also the importance of defining predictive biomarkers, in tissue microcompartments using different spatial technologies. So later in the year, he's able to apply this knowledge and studying using spatial technologies in a couple of different cancer types and published a few more papers. But what was also very interesting about what he was able to do is that 2020 hit, the pandemic, it literally, truncated or obliterated funding for a lot of scientists. They weren't able to get into the lab, and this was felt globally. But Arutha, with his understanding of the application of spatial, pivoted his focus to COVID-19. And through his collaborations with immunologists, respiratory physicians, and virologists, was able to run a pilot study using our technology and others to study COVID-19. And the results from that study have led to the identification of a signature in COVID-19 that can stratify disease severity. And Arutha may be talking about that today, but thanks, Arutha. This has been a great collaboration. So now I'd be remiss if I didn't provide a brief introduction to how nanostring technology works. So on the right-hand side here is our Chief Scientific Officer and Senior VP of R&D, Dr. Joseph Beecham. He looks like Albert Einstein, so he immediately has credibility. But all kidding aside, he's a fantastic and tremendous scientist and visionary. So he worked in collaboration with Dr. Gordon Mills, formerly of the MD Anderson Cancer Center in Texas, to conceptualize our platform and how you can identify with unlimited plex a number of targets in tissue, be that proteomic or transcriptomic. So this is the first conceptual drawing that he drafted on the back of a hotel memo pad. And what he designed or conceptualized was a series of antibody probes and or ISH probes that are covalently linked to a series of barcodes. And then using UV light, he's able to shine that UV light over areas on the tissue and liberate these barcodes so we can detect and analyze different targets. So as I mentioned here, we have targets, probes for protein and for RNA. And what's unique about those are these DSP barcodes that are covalently linked to the probe via this UV photocleavable linker. And that led to the discovery or the development and engineering of our GeoMix digital spatial profiler, which is what Arutha used and Dr. Rim used earlier. So moving into the future, we're starting to see spatial technologies expanding their profiling capabilities to address specific questions that are coming out of the lab and out of the clinic. Namely with technologies that can image tissue and profile genomic data at a cellular or subcellular level. So Rupert also mentioned this. So this is a low throughput methodology, typically when you're looking at single cell or subcellular resolution. And these images here are taken off of NanoString's spatial molecular imager. So this will be a platform that'll be released in 2020. So this can plex up to over 1,000 plex targets for both protein and for transcriptomic data. And then, of course, these technologies are going to produce massive data sets. And the future for AI, machine learning, and deep learning, as Rupert mentioned, and also as Quan will talk about, are very important to being able to interpret these data sets and the data in new and exciting ways. And there are a lot of players in different technologies that are starting to move into this space. So finally, just to cap it off, just to provide you with an indication of the overall market potential of spatial biology. So leading indicators suggest that the market will reach 12 billion over the next 5 years. So as you can imagine, there's a myriad of companies are entering into this space, and they're going to be providing unique and novel solutions to address those questions that I highlighted earlier. So it's a very exciting time and space to be in. So the market itself seems to be split between two platforms, one that can provide multicellular high throughput analysis, just as where the genomics sits, so up to whole transcriptome analysis. And then of course, as we move down to single cell and subcellular resolution, there'll be more of those imaging platforms that I mentioned, and our spatial molecular imager will sit there. And those really sit around, anywhere from the 100 to 1,000 plex environment. So that's my last slide for the day. So thank you very much, and I look forward to answering any questions you may have. Thank you.
That was great, Marshall. Thank you very much for giving all of us a glimpse of what's happening in the area. And this is a very exciting development, and all of us are looking forward to both working with the technology and then how it can translate. So we'll get back to some questions later, but now I'm very, very pleased to introduce Aruta Kulasinghe. I've known Aruta for many years now. He is an NHMRC research fellow at the TRI, Translational Research Institute, and at QUT. And I have also had the pleasure of witnessing Aruta's rapid rise over the last 5 years. So when right from when he just started his PhD to where he is now. So very, very glad that he is in this panel. Aruta is a passionate cancer researcher and great collaborator. He's an expert in spatial biology and liquid biopsy. Specifically, his research in the PhD time, he studied CTC, or circulating tumor cells. He used some state-of-the-art methodologies to isolate the CTCs, and he has made some strong contribution in head and neck cancer and lung cancers. We actively see that he's speaking in a lot of conferences, local and international conferences. And finally, I guess I can say this, he works more like 24 hours a day. So with that kind of introduction, Aruta, the floor is yours.
Perfect. Thank you, Shiv. Thank you for the invite. It's an incredible meeting to be a part of. On my slides, okay, Shiv, can you see them?
Yes. Please go ahead.
Thank you. And I think it's really,
One second, sorry, Aruta, you need to do a full screen. We are also able to see the next slide. So
okay.
Is there any way you can change it into full screen mode?
Can you see that?
Yeah, this is much better. Thanks.
Okay, perfect. Thank you for the introduction, Shiv. And yeah, really, really excited to be a part of this meeting. It's really timely and right after the AGBT meeting. So I don't think you could have chosen a better date for this meeting. So I'll talk about some of the work we've done in the last 2 years or so in the spatial biology space, how my focus has been on cancer, but we've really rapidly applied this to COVID-19, and there's some exciting data that's coming through. So hopefully I'll be able to talk about some of that work. So again, my work's in the immuno-oncology space, where we're really trying to identify predictive biomarkers of response to immune checkpoint therapies. So these drugs aren't cheap. They typically cost about $150,000 per patient per year, so it can't be given blindly. It works in about 50 to 40% of patients. So typically this work is being led in melanoma, head and neck, non-small cell lung cancer. And again, a subset of patients seem to do really well. You've got about a 20% cure rate in non-small cell cancer. So really identifying which patients will respond to immunotherapy is a key unmet clinical need. So this was at a AACR meeting 2 years ago, a photo that I took of Jim Allison, and just sort of made me think about meetings that we could be in person before COVID and the nature of meetings now. So I think I should have shared some of these slides with Marshall, or at least cross-check some of them with him beforehand. But anyway, so 2 ways to really understand, or 2 biomarkers to really understand predictive response to therapy have been the tumor mutation burden, and this has been known. There have been tests on the market from multiple different companies, from Foundation, Roche, a number of companies have these bulk sort of assessments of tissue. And I really or at least my lab's been really focused on understanding the tumor microenvironment. So I won't go into this image because Marshall's explained it, but I'll give an example of why this is important. So this is where two different tissues which have similar tumor infiltration scores, and the one is hot, but tumor is excluding the immune cells on the left, but the one on the right has a high degree of immune infiltration. So really understanding and being able to visualize this, the ability to overlay your pathology data with your transcript data or your protein data is really, really important. So at a bulk assessment, using a TIS score for this, you'd get a similar assessment, but being able to really visualize this, you can see the likelihood or predict the likelihood of outcome to a certain therapy by visually seeing where the immune cells are, whether they're infiltrating the tumor, and if the correct type of immune cells are recognizing the tumor. So on a bulk measurement, you wouldn't be able to make this call, but on a visual assessment, you could. So this is the power of this tumor microenvironment spatial profiling. So the technology we've used predominantly in the last 2 years has been this nanostring digital spatial profiler, where you typically, the beauty of it is you can pull out an archival block that's been sitting in a path lab for 20 years and section it and be able to interrogate RNA and protein, which isn't as readily available on a number of other competing technologies. So this was our preference of choice because we didn't have access to the fresh material, and you'd have to prospectively collect that. Whereas, you could walk into any hospital around the country and be able to access clinical trial specimens. You could access a whole lot of tissue that's archivally been banked. That's some work we did for COVID. And this material was shipped at room temperature around the world. And we were able to really get strong RNA signal off that, which was really promising. So you're able to stain a single unstained tissue section for your protein or your RNA of interest. You could typically do up to about 100 proteins. Above that with NGS readout, you can do between 96 to the whole transcriptome by looking at the RNA. You define your regions of interest. A UV light is shone above this region of interest to liberate the UV photocleavable oligos, and these are digitally counted. So you get expression data for different regions within that tissue type. So you're really able to walk across the tumor and understand that. So this is just an example of what that looks like. You can look at different expression profiles across different regions of interest, and not just in the PD-1, PD-L1 space. You can start to look at markers. You're able to look at about 100 proteins on a single slide. If you were to do this using traditional methods, you'd need 100 slides to get this data. So this is an example of a head and neck case where we could identify in 3 different tumors, VISTA, STING, IDO1, differentially expressed, but within that gambit of those 100 proteins. And when we look at, this is again head and neck patients, the type of response to therapy. So this is measuring non-progressive versus progressive disease in patients treated with checkpoint inhibitor therapy. CD8 counts weren't predictive in that cohort. So we did find that there were a number of other markers that are associated with a progressive disease profile. So CD44, STING, a lot of immunomodulatory molecules that we're looking at a great depth. This is a really preliminary experiment, but it showed proof of concept for this work. In lung cancer, we've done a lot more comprehensive work, and this is ongoing. This is all unpublished data where we've been able to look at second-line treated IO patients where we use a strategy of tumor masking. And what this allows is, because you use a UV light to overlay on this, what you can see is on the left-hand side is a tissue microarray, and this has been segmented based on cytokeratin. So when you shine the UV light above the tissue microarray core, you can liberate the cytokeratin positive signal in its entirety, and then interrogate that for your 100 proteins or your whole transcriptome. And when you do the inverse, you get a tumor microenvironment mask or the inverse of that. So you typically get 2 different signals, and that can be used to generate 2 different signatures per TMA. So this is a way of really looking in a large cohort, a really useful way to interrogate multiple samples in one set of lung cancer tissue microarrays. And then we've obviously done some orthogonal validation. So we've done some vector polaris work. We're working with the Koya Biosciences to access to 40-plex IHC, IF workup, where you're able to really add a whole slide assessment. So nanostring does regions of interest. You can do it more comprehensively, but it's a bit more straightforward using these other sort of lower cost methodologies where you're able to profile the entire tissue at a single cell level, and you're able to get the expression profiles at a single cell level. And what you can see is on the top left, this has been annotated. It's been broken down into single cells, and then you get the expression profiles of those individual cell types. And there's no spectral overlap. And that's one of the beauties of the Polaris system. So you're able to do things like distance mapping. So at the bottom here, these are just 2 cases of the non-small cell lung cancer TMA cores, which have been stained for this 7-plex cohort. So now you can start to ask questions from that from the core about the composition, the distance mapping, how how far are your CD4 cells from your tumor, the CD8 cells, and start to do neighborhood plots and sort of clustering of those data sets. So I think where this is heading is, this is some work with Enable Medicine, which is a team out of Stanford, is where we're hoping to move from this tissue multiplex IF image to these complex clustering maps where we're able to really understand what's going on at the cellular level and understand the links of each cell type to its nearest neighbor. And I think this is going to be a really, really powerful tool when we start to overlay these data sets. So overlaying the RNA, the protein, the mutational data, the exome sequencing of these tumors, and really get a multi-omic appreciation for what that is. And then try to inform down to an H&E if that's possible, is one of the goals. But this is some exciting work where we're hoping to really move rapidly in this space and validate a lot of our nanostring data sets. With that, I'll just touch on some of the spatial work that Marshall talked about. We've now done a lung study looking at autopsy samples from COVID-19 patients collected in Brazil. We've carried out a myocardial project and a placental project too, and we're about to receive COVID brain tissue as well. And it's been a fascinating study. When you stain these tissues with RNA scope, the lungs lit up like a Christmas tree. But then when you looked at the myocardial, the placental tissue, you couldn't see the virus present by RNA scope, even at the transcript level. So we think it's a multi-organ inflammatory response, but you do get localization to the lungs. So what we were able to do in this project was to, we had the tissue microarrays. These were H&E stained for H&E stained Mason's trichome for fibrosis. We had the IHC for the virus, RNA scope, PAT review. So in Australia, one of the real challenges was to find a pathologist that could look at COVID-19 lungs, because it wasn't, when we called all the hospitals locally, no one had seen COVID-19 lungs. So we had to reach out to some collaborators to access this tissue. So one of the key findings of the study was a number of type 1 interference response genes were upregulated in COVID versus control. And that same gene set seemed to be upregulated in COVID versus H1N1. And we've done some further analysis of this IFI27, and it appears to be a triage biomarker for disease severity. So when you measure that gene at the pre-symptomatic stage, day 0 to day 2, you can predict how severe the disease symptoms are going to be or likely to be. So we've now looked at 3 different cohorts from Brazil, Chile, and Iran, and the data seems to match up. So this is some work that's ongoing, and we hopefully, it's been enabled by spatial biology. It's a purely discovery tool, but this was one of the interesting findings of that study, and this has now been validated in the blood and nasopharyngeal swabs of patients. With that, so that's just a preprint at the bottom of that paper, and thank you to the patients that have contributed to this work and the collaborators across multiple different tumor streams and COVID-19. With that, I'd like to take any questions. Thank you, Shiv.
Oh, Aruta, that was terrific. Thank you so much for sharing all of this. And, this is so important for the audience of this conference. And this will mainly be inspired by a lot of future postdocs and PhD students and research folks, not only in India, but the whole subcontinent. I think this talk gives a sense of what's happening at the moment and where do you see this future of spatial biology. And with that, it is let's go to the last speaker, and it's a great pleasure to introduce Kwan. Uh, um, Kwan was introduced to me by Aruta, and then I started looking at some of his papers and his work. It's just excellent to see such a big contribution in such a small amount of time. Before I go into what his work is, just very brief introduction about Kwan. So he's currently group leader at Institute of Molecular Biosciences at the University of Queensland. He's leading a genomics and a machine learning lab. And their focus is on neuroinflammation and cancer immune cells. So he's an expert in single cell data resolution or data analytics. And he has a PhD from UQ in 2013. And then he went on to do a postdoc at Riken in Japan. He returned to CSIRO as an OC research fellow and then subsequently IMB. So he's currently on a prestigious ARC DECRA research fellowship. And as I was saying before, he has already developed many innovative machine learning approaches to find cancer cells and predict their progression within histopathological images. And they are one of the pioneering labs in India and globally and emerging in imaging omics approach. We are very excited to welcome you, Kwan, here. And with all of this, eventually the most important bit is also the data interpretation and the analytics. So I think it would be fantastic to see how you're coping up with so much data and what are your plans moving forward. Kwan, over to you.
Thank you very much, Shiv, for the very nice introduction and for having me here. It's great to listen to all the talks today, and I'm learning a lot, and I really look forward to more collaborations and new collaboration with companies like the one from Rupert, and yeah, hope to be connected after this seminar. So I've just shared my screen. Do you see it in the full screen mode?
Yes.
Okay, yeah. So I will go very briefly about some of the approaches that my lab is doing to diagnose cancer at two aspects. First is the high resolution, so the single cell resolution, and the second aspect is at the tissue context, because we know that cancer is as a whole as a heterogeneous combination of the cells, and how can we capture both a single cell resolution and the complexity of the whole tissue is the ultimate aim of my group. So let's have a quick think about the landscape of the traditional pathology now. So basically, like from any cancer patients, usually we have a tissue block from the patient, and what happening is from that tissue block there will be tissue section, and the tissue section will be stained usually at the gold standard. That's a H&E staining that tells the nuclei and the cytoplasm. And then the doctor may request for a follow-up validation by staining with another protein markers. In this case, I'm showing an example of colorectal cancer, and this is the H&E staining, and this is the P53 marker staining. Now, the AI community are really active and innovative in this space, and people know how to now use thousands or hundreds of thousands of images like this with the combination of pathologist annotation to classify the image and then train the image and enable the computer to look at a new image, H&E image, to classify where in the tissues are cancer, in this case the red color, and where the normal. Now, in our team, we came up with an innovative approach here where we could combine the P53 as a stain marker with the H&E marker into one data set. That means that we molecularly label the traditional H&E marker with protein markers, and that allows us to create a data set where we can really train the machine to look at every pixel in the image and classify it into cancer versus non-cancer. So it's one minor way of us making a little advance in the space of the computer-aided diagnosis, where we combine the molecular information into the traditional histological image and do the classification. Now, but we thought that we could do better than this. And that is when the spatial technology come in. So if we recall in the previous slide, I show one tissue block, two tissue sections, and integration of the data between these two tissue sections with one protein marker. So now with the various spatial technology here, I'm just showing one example of the technology. We can do on the same tissue block, get the H&E image and measure 22,000 genes in that image. It's like we have a 22,000 tissue sections that we can integrate with a H&E image. So that really opened a huge potential for us on how can we utilize the molecular information to inform the traditional pathological image space and develop some advanced computational methods to improve the diagnosis. Now, here I'm showing the breast cancer tissue here. From this tissue, we get the gene expression data, and also we get the imaging data. Here, I digitalize that imaging data by the pixel density value as red, green, and blue channels. And importantly, for this one single tissue here, we could measure 3,000 spots. And in each spot, we have one measurement like this, about 5,000 genes, and the same measurement for that exact spot with the pixel information. Now that create a training data set with really big size. If we consider one spot as one data point, with one tissue section, we have 3,000 data points, and that's really good for machine learning algorithm. Also, we can make quite simple comparison between what we can learn from the data we get with what the pathologist can tell us traditionally. Now, with this breast tissue section, the pathologists in here, actually, we combine the result from 3 pathologists into one, look even more carefully than what's been happening in the clinics. So 3 of them look at one tissue section and give us different classification of the tissues. Now, as you see, the number of cell types and cell states in here is still quite limited. Now, with the information of the gene expression in that same tissue, we could find many cell types. And in fact, we know that in here, one color, one cell type. And we show that each cell type that can be identified by our analysis approach actually make biological sense, because when we look at the gene expression here, for example, here we see the red color. That means where this particular gene is expressed highly, match very well with this particular cell type. And this cell type here is just one part of this broad classification of the pathologist, which is the invasive carcinoma. So that means that from one region like this, we can find many subtypes based on the spatial data that we get. Now, our task in here is how can we automate that process? How can we make use of the data to create analytical products that may be useful for the pathology practice? Now here I'm showing one example of a software that our group produced. The name is Spatial. And in this software, we apply neural network, or people also call it artificial intelligence, to combine 2 types of data. So the imaging data here and gene expression data here. And there are various architecture of that network that we can use to solve a separate task. For example, the task here we are showing is to classify in prostate cancer, the cancer cell versus normal cell. I'm gonna go into this example in more detail later. And we can also look at the disease stages. So here we look at the one of the neuronal disorder disease, and there were 4 stages in there, and we could apply neural network to classify each of the 4 stages in quite high accuracy. I'm gonna go into a bit more details about our approach on how to use this particular spatial transcriptomics data set to train the model, the neural network model. So let's look at this as one example of the prostate cancer. And as I mentioned before, we can split this image into many smaller images to increase the sample size. So for each smaller image, we call it a tile. We can use the standard image computer vision approach, in this case convolutional neural net, and we did apply some small tricks like transfer learning, where we can really make use of the huge imaging data sets out there that are not related to medical field, and apply that model to extract the useful information from the cancer image. And from there, we can extract the high-level representation of the image. In other words, the numbers here can capture the pattern in this image in a way that we can apply those numbers to regenerate similar image like this. So that means the magic or the power of AI in here is really powerful in the way that it can turn a very abstract image in here into numerical values, where we can integrate it with the traditional genomics data. In this case, that's RNA sequencing data. And by doing this, we can really combine the imaging data with the sequencing data into a neural network model. We train the model and we do the classification of cancer cell versus non-cancer cell. So this is, again, the prostate cancer. And we see in this red area here is where the pathology is determined at the cancer region. But with our approach, we could really improve the resolution a lot by looking at the entire tissue and predict every dot, whether that dot is cancer or non-cancer dot. And that obviously will increase the specificity of the classification a lot. But interestingly here, what we show is, we show when we combine both gene expression data and imaging data, we could increase the accuracy and specificity of the prediction compared to using one type of the data. So really this spatial information plus the H&E image can bring in new capability for us to automatically look at the H&E image and at the high resolution predict where in the tissue is which cell type. Here I'm just doing the cancer versus non-cancer, but actually we can also look at immune cell versus cancer cell like in here. So this is pathological annotation. You can see this small part here that's identified by the stroma cells. And by using the computer without knowing anything about cancer, we can actually identify that region as the stroma cells like in here. So you can see with the combination of gene expression and then the imaging data, we can predict the very high accuracy and specificity. Now, before I finish, I also want to show that beyond diagnosis or classification, we can also make some prediction that may be useful for the prognosis. That means that from one snapshot of the tissue at diagnosis at the time where the patient came to the clinics, can we predict if that image will tell us that this patient gonna have a metastasis or not? And this type of multi-dimensional data allow us to do such a thing, because by using the transcriptional profiles of 22,000 genes, we can look at every cell types. And for every cell type, we can estimate the transcriptional state of that cell type. Now I'm showing this is a non-invasive cell type, and this is the invasive cell type. we can connect the non-invasive to the invasive cell type in an inference way, so that we know that if this person in a year time, we get another tissue section, we will see something like this. So that is a way that we can build what we call pseudo time. That means an infer time from one snapshot of the tissue, but with a lot of heterogeneity within that tissue, and we borrow the information from that heterogeneity to predict the future of the cancer patient. So that is the idea. And in the summaries, I've shown you one aspect of the work that our lab is doing, and here's the tool software that we produce that can be very helpful to do something like this. So like we like to combine the spatial transcriptomics with the single cell resolution. So this gives us the cell type information. This gives us the location and the interaction and the dynamic between cell types and an ecosystem. And then this AI come in that allow us to make the best use of that data set. So I'm really hoping that we can bring this kind of research to the clinics sometime as soon as possible, and it's our main focus now. So I'm very happy to answer question and to the discussion section after this. Thank you.
That's great, Kwon. It was a very, very interesting talk. And the last bit was very exciting to see how you can possibly predict the patient pathway into metastasis or not by those imaging. And I must tell you, India has a huge imaging expert skill set. So there are these institutes called Indian Institute of Technology, where we have a lot of imaging experts, and I'm sure something like this will excite a lot of people. So I have really two ways to take this conversation forward. One, I have a set of questions that I want to ask some of the panelists. And second, really, I would allow the interaction between the panelists. Given that everybody is such an expert, I would like to start with the second one rather than the first one. I have some context related questions. So does anyone, like, can someone start asking a question to a different panelist? How do we do this? Because everything is so exciting. I'm not really sure where to start in terms of whether it's analytics or the future. So who do you want to, like, can somebody start a conversation? Yes, Kwon, please.
Yeah, so yes, I may start with asking. I dash. Sorry if I pronounce your name not correctly, but your talk really inspired me in the way that you can really bring this this kind of technology to the rural area and to increase the access to many people. And I believe that with more users, more information, the technology will definitely keep improving. So could you elaborate a little bit about whether you are working with some research institutions and you think that what happening in academia may help a bit with your work at the moment to bring some of the new methods to improve your work pipeline?
Yeah, thanks, Kwan. Yes, definitely, Kwan. I mean, we've in the past worked with a few academic institutions here in India. Some of them being the Indian Institute of Technologies that Shiv was mentioning about. A couple of them. We also worked with the Indian Institute of Science, IISc, that is based in Bangalore. So together with multiple components that we put together, we worked with them in separate areas, and finally we were able to synthesize all of that into a product that's now being put out there. And to the second part of your question, Kwan, definitely it makes a lot of sense to continue this collaboration, not just within India but across borders as well, and then take some of this cutting edge technology to the front line out there. because finally these are the technologies that can act as force multipliers and kind of help penetrate the market better, help increase the social impact. So I'll be very, very happy to talk to you further and explore any synergies that can come by Quant. Of course, we'll have to figure out how to make that into a point of care setting, or how it can be able to fit into a price point that makes sense for the market. If not, we'll have to look for a segment that to which the price points make relevance. Right. So that's definitely something I'll be excited to discuss with you further on.
Mm-hmm.
That's great.
Yeah.
That's excellent. So, I actually have a great joy in connecting people, and when something like this happens, and the whole purpose of this forming panel and talking about it is so because just imagine the potential if Quan and others start to work together, the very, very similar field. So I will start with one question. So this is again to Rupert. So Rupert, where will you be investing in the future in the tissue agnostics, or in other words, where would be the future investments for tissue agnostics given the changes and the new innovations that you see? Rupert?
Yes, I can hear you. Thank you, Chief. The field, I think this discussion has shown how exciting the field is, right? And for me, as a biologist, as I was originally trained, but also as a businessman, it's really, I almost want to call it a pleasure to work in this area and with people like you. It's also, for me, an opportunity to realize my dreams, as a company, to provide tools which help others to expand the knowledge of humans. This was when I was a student. I was once asked during a job interview, what would you really like to do? And I thought, what would I really like to do? And then my answer was, I would like to help others to expand our knowledge, right? On a global scale. Not only work myself on a certain molecule or one disease, which will also expand our knowledge, but to provide a technology which helps others to increase our knowledge. And this is also where we as company are going, the direction. As I said, the focus is on machine learning, on the future. I briefly showed the slides, the virtual histologist. This goes in the direction what also Quan has presented and what the other panelists are doing to support. So I think we need computer assisted diagnostics. We are approaching a level of knowledge of data that needs to be integrated in a decision process, which goes beyond the capability of the human mind, right? So, and especially when we are talking about this molecular profiling, precision medicine, decisions taken on in the basis of an individual patient, the situation which is present in an individual patient. You cannot do this anymore statistically, right? So we have a number of development projects in our pipeline. Some of them are still confidential, so I cannot disclose them. But it all goes into this direction of more automation and computer-assisted analysis for the researchers. So Adarsh has made a clear point of this. It has to be easy, and this is a challenge. at the same time, when we see how knowledge is expanding, how more and more factors and molecular or genetic cellular factors have to be integrated in a diagnostic decision process. And this is on the one hand side. And on the other hand side, we have the situation like you described it, Adarsh, in India, in the rural areas. Hundreds of million people do not even have, cannot even measure their blood pressure properly, let's say. Yeah. So this is a big gap that we have to bridge. And this can only be made when we develop technology which is easy. Yeah. So easy to use, more powerful, but also easier to use. Always more and more complex is not the solution. Yeah.
Yeah, that's
So
great.
this is our main focus in tissue gnostics. Yeah. Investment in terms of geographically, we have actually started, as you said, we are a European company. We have started in Austria. Then already 2007, we founded a subsidiary in the US. 2017, we founded a subsidiary in the Asia in Pacific region. Now working here currently in Australia on the Australian division. And then now we are having projects underway in Africa and in South America. Yeah? So these are, and India as the subcontinent, and with what you have seen, what you have presented, Adarsh, this amount of people and this potential, yeah, the market potential that is there, not this year, but over the next 50 years. Yeah. This is incredible and will also for sure require our attention.
Absolutely, that's very well said, Rupert. And I can also speak from the kind of student cohort that I have. You know, it's just not India, Sri Lanka, Bangladesh, Nepal, Bhutan, Pakistan, the whole, the subcontinent, I think will have very similar health challenges in the future. And everything that we develop and we translate, we have to somehow think about how globally this is applicable. You know, we have Medicare here, we have systems in place that allows us to work in a different way. But having worked or working in the indigenous space, there are some challenges in the indigenous health space which are very similar to what Adarsh explained, the rural Australia where the indigenous people live. And I see some of the applications of what we discussed today to some part of Australia as well. So that's a comment that I would like to make. And anybody else wants to?
I have a question, Shiv, if that's okay. Can I?
Yes, others, please go ahead.
Yeah, to Rupert, Quan, Marshall, and even Arutha and US as well. So one of the hot topics, I'm sure all of us in touch with what's happening out there, and when it comes to AI in healthcare, has been two specific parts, right? One is in terms of explainability to the clinicians, because they don't like the whole concept of a black box, right? And they completely abhor the fact that we don't know what is happening under the hood, and therefore we can't trust it, right? So that's one question I want to hear your thoughts on. The second part is in terms of the biases, right? The biases that are creeping in the data sets, and therefore the downstream impacts of those biases in your data set. So I'm kind of keen to understand how the successful businesses, Rupert and Marshall and Quan, yourself, how are you guys all handling these two aspects? Because I think this very well could set the tone for how much of adoption from the community, right, from the clinical community will happen over the next few years, and therefore also the kind of impact that we will be able to penetrate the markets with.
So I would say, if I may answer
first. Like to hear your thoughts.
Sure.
As we are here in this group, we are working on applying artificial intelligence to translational questions. I think it is very important, and I can only speak for myself, for tissue agnostics. We are collaborating with academic institutions in the field of machine learning who actually first develop technologies, novel technologies of machine learning, but and this is second, most importantly, also work on understanding why a certain model gives a certain result. Yeah. So I can understand the concern of pathologists or of anyone who is afraid of a black box. You put something in on one side and on the other side, something comes out, but you do actually not really understand why it comes out. This is what we have.
Exactly.
Also currently, when you look on current support vector machines, some of them have thousands of features which they consider, and then you get a result. This result is very reliable, but sometimes then an unexpected result comes out. And this is exactly the black box aspect. Yeah. So my message is it's very important that we from translational research, where we try to apply a technology to a clinically relevant question, that we also closely collaborate with the basic technology institutions. And this is one reason why I'm at QUT here, to understand what is really going on in that black box and to what level we can trust it. Yeah.
Yeah.
Excellent. Excellent. You want to respond, please?
Yeah.
Yeah, so I agree with Rupert a lot about the interpretability is a very active research area at the moment. So AI 3 years ago is quite different to AI now in terms of interpretability. There's a big improvement there. Our group, we work on that as well. For example, from the H&E image, we can map back to the image to tell which part of the image that really makes the model predict that this is cancer or not cancer, whether it is because it's the densely populated nuclei regions, or whether the nuclei is a bit bigger or smaller. So that's an active research area. And secondly, I think the technology itself also helped to improve the interpretability, in the sense that we now integrating molecular information into the image. So it's not just pure image, but now we know that this part of the tissue, these genes are expressed highly and the other genes are not expressing. So by incorporating the biology and the machine learning in here, we can really improve our understanding of the model itself as well. And the third one I like to emphasize is we're developing computer-aided tools. So these tools would not replace the pathologist, but it helps the pathologist to do their job better and faster. So at the end, we would still need, I think, pathologists to validate what the computer predict. Yeah.
I think, from my experience, and I think it echoes what Rupert and Juan have said, is that, you need to have the clinicians in those discussion rooms where you're going through the experimentation, where you're doing the data analysis. And if you have those multidisciplinary teams formed where they're part of, from the concept of the project all the way through, and you bring them in on meetings, especially the pathologists. So for the COVID work, they're an integral part. For the lung cancer work, they're an integral part. They're annotating the tissues. They're going cell by cell by cell, telling us what's there. So having that integration and having them present in those meetings is really important. So you're not just getting tissue from them, but they're part of the conversation throughout the project. And then, they're closer to the data. They know what you've sort of done with it. And, it's not a foreign thing to them. So and a good example is the vector Polaris actually has, so on multiplex immunofluorescence staining, it's got a view called pathology viewer. And I always, it baffled me, what is this? And it would just brown stain your image. And all that was doing was taking your IF image and making it a brown stain to make it friendly to a pathologist to assess. So even, the technologies know this, and that's why they've designed them within them. So I think, really making sure those clinicians and the pathologists are part of your discussions early on and through your projects really important.
Yeah, there we are touching the field of, recognition psychology. Yeah. In fact, we also introduced a feature which we call pseudo IHC, where we convert a fluorescence image so that it looks like a IHC sample. And this is actually based on an experience that I made years ago when I showed a DAPI, so a fluorescence sample, to a pathologist and asked him, is this a tumor? And the pathologist said, I don't know. I need to see the hematoxylin image. I said, what's the difference of showing blue on white or blue on black? When I show the pathologist blue on white, he's able, or he or she, the pathologist, is able to recognize whether this is tumor or not. But when you see it blue on black, he didn't know. So this is about perception, how human perception works, right? It gives you seeing a dark blue on a black background gives a completely different, literally a different image also in one's mind when you have for decades looked on blue nuclei on white, right? So this is the other aspect, the human how the human mind itself works. Also, this has to be reflected when we deal with computer-assisted methods. It's not about the methodology alone. It's also how the result of a methodology is perceived by a human operator.
Correct. Yeah, it has to do with, I believe, with your level of comfort. Yeah, the level of comfort of different groups. So, in research or pathology or anything, we're siloed, right? We're very good at one specific methodology. That's what we know. We know it very well. Anything outside of that comfort zone can be, somewhat scary. But as Arutha said, and I think as research is moving forward, we're starting to collaborate with different groups. You bring them into the fold, and we're starting to integrate and understand those technologies. I also think benchmarking against gold standard methodologies that certain groups are familiar with to show that your platform is actually performing and giving the exact result is another thing that's very important as we move forward.
Yeah, and I think Marshall, David Rim's a pathologist by training, and the fact that he's using all these technologies is really impressive. So I think it's at that translational space where the pathologists are really seeing the value in it, and I don't think it's replacing them; it's complementing their workflows.
Yeah. So we are sort of running out of time, but I just want to ask whether all these developments have changed anything in the medical training. So when, the radiology training, what they've originally trained for the last many years, is there a new course on how AI helps and what is the black box? Does anyone know how the medical training is changing? Because the uptake that we are all talking can be trained very early on when they are actually doing the medicine. If not in the MBBS, but the specialized, radiology training and really working closely with some of these new developments in the last 5 years. Is anyone aware of such new changes in the curriculum of radiology?
So I may just give the case in my group. So we do have a medical doctor student who's working on the writing scripts and code to train the machine to recognize the images. And definitely there's a valuable combination with the medical background plus the coding skills. I also heard that the UQ, they do have a plan to introduce the data science course to the medical training. So it's looked quite positive to me. Yeah.
Okay, excellent.
So what I have seen in Europe is that actually such new technologies are incorporated in the curriculum. And for example, the European Union puts a lot of effort in training early stage researchers. Like what I have briefly shown in one slide, these 5 European innovative training networks that Tissue Gnostics participated in, they were all focused on this one goal. train young researchers to the latest state of the art in science, including machine learning or whatever. Yeah. This is the so-called Marie Curie program of the European Union, which focuses on bringing the latest knowledge into the minds of young researchers.
Okay.
But Shiv, if I may ask one more
Yes.
topic, or offer one more topic for discussion, because it relates also to what you stated about the training, the need for training and update of curriculum. I think one thing that also the corona pandemic has made very visible is that there is a gap, and I would say an increasing gap in the development of technologies and the clinical implementation. And this is also what we have addressed earlier, in a way that technical development is much quicker than the clinical implementation of a new technology. And this also relates to the definition of regulatory constraints. Like, you know, in order to be allowed to use a vaccine for corona or a new AI-based diagnostic technology for cancer, or just one type of cancer like prostate cancer or breast cancer, usually takes many, many years. It requires clinical studies, approval by FDA or other regulatory authorities in individual countries. So we all here at universities, we put a lot of, or also in companies, we put a lot of energy and money into research and development to create new technologies. But then the step from having a technology ready to its clinical application is even bigger than the actual creation of the technology and costs way more money. Yeah? And we have seen with Corona, the vaccines, normally it would have taken 5 to 10 years to get these new RMA-based vaccines approved. Corona forced us now to do it within a year or less. Yeah.
Yes.
And I would like to hear what my colleagues at the panel think about this.
Others, you want to have a quick response for that? So, because you have got your compliances for your innovation through the Indian system, do you have any comment for this?
Absolutely. Rightly said, Rupert. I think the old paradigms of so there's a certain kind of urgency that's coming into the whole the bureaucratic system, which deals with the approvals and the regulatory pathways. So what we've seen also is not just, of course, COVID was the catalyst, I would say, but I think that has crept into the system where we've seen approvals not just for COVID specific things, which are of course fast tracked, but even other healthcare initiatives are being taken up much more seriously. And Shiv, as I was mentioning to you during our earlier discussions, that a lot of approvals are happening in a much more streamlined fashion, even in India, which traditionally was known to be a little bit more bureaucratic. But now I think there's a market change, and it's happening much more quicker, more streamlined. It's all online. You kind of apply, you provide all your data documentation online, and it happens fairly quickly. And I think that's slowly creeping into the rest of the ecosystem as well. So that's a welcome change that I have seen in India happening.
Sure. That's great. Yeah. So I have a question, probably maybe just one more, but in the conversation, so to Marshall. So see, nanostring is an expensive technology, and there are two ways in which this can work. Given that my own building has one of that, and thanks to Aruta and others to bring that instrument, how do we take this to Indian subcontinent? So for example, do you recommend center of excellence type of models where we have a few of these in the really key top tier research institutes where you can build the skill and then others can use it? Or do you have a model in which you can cater to hundreds of different research places that are there in India.
Yeah, definitely. I mean, certainly our goal is, our dish, it's that is we want to democratize access to genomics as much as we possibly can. So whether or not that's, placing within specific research institutes, within CROs that can provide a service, for a certain price, et cetera. Those are some models that we put in place, and we have some systems that are going into India. So I do see that happening in the near term. In terms of cost saving, I think one of the interesting aspects of our technology, what researchers can do, and what Arutha highlighted in one of his studies, is that you can use a tissue microarray. So you can place 48 individual core samples across a slide and run whole transcriptome on those samples. So if you divide that by the cost per slide, it actually takes the cost down on a per sample basis to something that's actually quite manageable. So that may not be possible for all studies. I understand that and the tissue type that you're working with, but that certainly is a way for a cost benefit way for groups to approach the technology. Okay, thanks for that. And then of course over time I anticipate that prices will probably drop. Don't quote me on that, but. Yeah.
So I think we are really running out of time. It's so exciting to keep talking, but we can certainly take this conversation offline through the emails, etc. But again, on behalf of TechBharat and all the potential audience who are going to watch this live tomorrow or recorded session tomorrow in the conference, thank you very much, everyone. I really appreciate your time and sitting through this. And Shekhar, would you like to close?
Yes, sir. Extremely thankful to you, Doctor, for anchoring this excellent session. In fact, it was indeed very comprehensive technical deep dive session. I also thank your co-panelists, Dr. Kwan, Dr. Rupert Ecker, Dr. Marshall Patel, Dr. Amrita Kulasinghe, and Mr. Adarsh Natarajan. We have taken a lot of notes from this session, and we will be passing this to relevant government authorities. Hope they will take note of it.
Sure, I just have to add that because I didn't get a chance to say this. So this is a very important conversation. So a report is going to the Department of Health in India, the Ministry of Health in India, sort of take home messages from all of this that we're talking. So this is directly supported by the state and federal governments. And therefore, I was very keen to bring the best of the people, best of the panelists to this. So as Shekhar is saying, all of this will, with the courts and with our, some of the information will go to the federal government, isn't it, Shekhar?
Yes, Doctor. And on behalf of TechBharat, I once again thank the great panel. Thank you very much.
Thank you, everyone.
Thank you very much.
Thanks, everyone, for a fantastic.
Bye-bye.
Hope to stay in touch.
Thank you, sir.
Bye-bye.
Thank you. Bye.